Well, welcome to my next class on this topic. Yes, in previous class, we have discussed about artificial insemination, and I told you the different techniques like MOET, that is nothing but multiple ovulation and embryo transfer, or it is also referred as super ovulation and embryo transfer. As the name suggests, multiple ovulation. What do you mean by that? Here, what is ovulation? Right? Because ovulation is the process of releasing of, right, the ovum from the ovary is known as ovulation. Right? The process of releasing, right? The ovary ruptures, then the ovum is released. Such a process is known as ovulation. Now, why we call it as multiple ovulation? Multiple ovulation and embryo transfer. What do you mean by multiple ovulation? Means to say that multiple suggests means what? Multiple means many. Multiple ovulation here in this process and embryo transfer. Embryo transfer. These are two techniques combined together here in order to increase the size of the herd. In your final exam, it can be asked. The question can be asked like this. A few years back, many students got confused by this question. Right? Explain the technique used to increase the size of the herd in cattle. To increase the population, to increase the number of the cattle. Which technique is used? Multiple ovulation and embryo transfer. This is one small topic and this is another topic combined together to form a technique, right? In order to increase the size of the herd, to increase the population of the cattle, right? Cow, buffaloes, sheep, right? Horses, right? Mare, rabbits, the animals which are useful in agriculture and for other purposes. Such animals are reared by this technique. So here, normally ovulation takes place during the heat period in case of animals, during estrous cycle. Normally in mammals, higher mammals, only one ovum is released from the ovary. Such a process known as ovulation. Why we call them as multiple ovulation means, in this technique, the cow or the animal is made to release more number of ova. Hence, it is known as multiple ovulation and embryo transfer. The technique is known as MOET. It is also known as SOET. Instead of multiple here, the terminology that is used is known as super ovulation and embryo transfer. Clear? I hope it's clear. So, during this process, what is done to the cow, the steps, right? The technique I am explaining now. In the very first technique, right? The very first step, right? The cow, the selected breed cow is selected. That cow will be injected by LH and FSH. Now, recall these hormones in your last chapter of first year topic, right? LH means luteinizing hormone, right? And FSH, follicle stimulating hormone. The hormone that is responsible for the formation of Graafian follicle, responsible for rupturing of the ova, rupturing of the ovary to release the ova. Such a process known as ovulation. So these hormones are normally naturally produced in the mammal's body. So normally ovulation takes place, but here we are we are injecting these injections from external source. Externally, these injections are induced, right? One is known as LH, luteinizing hormone. The other is known as follicle stimulating hormone, FSH. These hormones are injected to the cow, so that this animal produces many ova or more ova. Right? Normally, one ovum is released, natural conditions, normal conditions, single ovum is released, whereas here this animal is made to release more ova, right, by injecting hormones like luteinizing hormone and follicle stimulating hormone. Hence, in this process, many ova are released. It is known as multiple ovulation or it is known as super ovulation. Then, when this many ova are released here, fine, clear. After that, they are in the second step. The semen, which has been already collected in the previous topic, we have discussed artificial insemination. The semen is collected from an elite bull or a selected breed or a very good breed, good bull, which produces quality semen, which has different characteristics, traits, right? High milk yielding or more meat production. Such animals, sturdy, heavier, strong animals, such bull is selected from the elite bull, right? By using artificial insemination, semen is collected. Cryopreservation, you know that in the previous technique we have discussed. Here also the point comes here. Then the semen is injected into the female reproductive tract, into the female reproductive tract, right? By using catheter, and these technicians or the doctors, veterinary doctors or the researchers, technicians, right? Before they inject the semen into the cow, by inserting their hands into the anus, they remove the feces, they remove the dung, cow dung, whatever is there inside. They remove that so that it should not be an obstacle during this process. Then, by wearing gloves and covers, they do it practically. After the removing of the cow dung from the digestive tract, right, from the alimentary canal, then the catheter is taken. Then camera is also it's micro camera is inserted into the reproductive tract of the cow. Then syringe also, like the catheter is injected, or till here they insert the hand through the vagina of the female of the cow, of this animal. Then they inject this artificial insemination is done, and here the fertilization is very much confirmed because the already cow is in the heat period where ova is released. Now the fertilization is very much confirmed here, conception is very much confirmed here, right? After that, when it is introduced, right, then it is allowed to undergo fertilization by natural process. When it undergoes fertilization, around the embryo is formed around 8 to 12 cell stage. 8 to 12 cell stage. For example, one cell, then it becomes 2, then it becomes 4, then it becomes 8, then like this. So when it becomes embryo, many cells are formed. Mass of ball of cells is what is known as embryo. When it is formed at the stage of 8 to 12 cell stage, these embryos after fertilization, what we call as embryos, are collected from that cow. Again, they are removed from this cow. From the which cow? From the actual mother, from the biological mother, from the genetic mother, in which the artificial insemination is done. Here. After fertilization in the reproductive tract of that cow, after fertilization, after embryo is formed, these embryos again, by inserting camera, then by hand till here, because it has to reach that level, then the ovum is collected. Here ovum is collected by non-surgical method. No surgery is done, no cutting is done. Right? The hand is inserted into the cow, and that small embryos are collected. They can be preserved. Two methods are here. They can be preserved by what method? Yes, by what technique? Cryopreservation. These embryos can be preserved by cryopreservation. Yesterday I have discussed. Cryopreservation, minus 196 degrees Celsius of liquid nitrogen is used, or without preserving. If it is required for further use, you can preserve, or directly these removed embryos. For example, if they have removed 8, they are differently injected into 8 different cows. 8 different, not injected, they are implanted. This process is known as implantation. The attachment of the embryo to the wall of the uterus is known as implantation. After embryo is formed in the surrogate mother, in the actual mother, biological mother, the genetic mother, embryos are taken from one cow. Why? Because in that cow, how many embryos are growing? 8 to 12, which is unnatural, which is not possible. This cow cannot lay 8 to 12 calf at a time. The system is not so well developed. It is developed for only for one calf or one young one. So here, after 8 to 12 embryos are developed from that cow in which artificial insemination is done. From that cow, these embryos are taken. They are either preserved or they are immediately implanted, transferred into other cows. How? By means of hands. And these embryos are taken and released into the female reproductive tract, where these cows are also injected with hormones, because implantation has to be induced there. So implantation takes place. Now in these cows, 8 different cows, for example, 8 embryos we have taken from here. In these 8 cows, these embryos will be developing after the gestation period of around 2 months, around 7 to 8 months, fine, or more than that in different breeds, right? Different gestation period comes. Clear? What is gestation period? The period of the time taken, or the duration from the time of fertilization till the delivery, till the birth, is what is known as gestation period. In case of human beings, it is 9 months. So for understanding purpose, I am telling you this. Here we are dealing with the cattle. Some cattle around 9 months, 10 months, 11 months, it differs. 18 months in case of elephant, it's like that. Gestation period, shortest gestation period in case of mice, fine. Pygmy shrew, 13 days. It may be asked in your NEET also. So in these 8 different cows, 8 embryos are developing. 8 embryos are developing. They undergo gestation period. Now this cow, which is the actual cow or the genetical cow, or we can say as biological mother. Why we are saying as biological mother? Because the ova belongs to this mother. Ova belongs to this cow. Hence, this cow is known as biological mother. Whereas these cows that are providing shelter to this embryo for growing, such cows are known as surrogate. Surrogate. These techniques you understand in 4th chapter also, human reproduction and reproductive health. There also it comes. Surrogate mother. What do you mean by surrogate mother? These cows are not biological mothers. These cows are only providing the shelter. Their uterus is being utilized for the development of these embryos. The embryos are this. The ova belong to this cow. The semen belong to other bull. But only here, these cows are used for the purpose of development of the embryo. The cows are the animals, right, which provides the shelter for developing, growing of embryos. They are known as surrogate cows or surrogate mothers. These cows are serving here. They are acting like surrogate mothers. They are served as surrogate mothers. After gestation period is completed, 8 cows or 8 bulls or cows, whatever it is, right? They are born at a time. And now, what is the advantage here? You may ask the question, why this 8 should be transferred to 8? Because 8 to 12 cannot fertilize, cannot grow into a young one into one cow. That's one thing. Second thing, this cow is genetically strong. So here these cows are used as surrogate mother. After a period of 1 month or 1.5 months, this actual cow or genetic cow is again ready for insemination. Artificial insemination. So this cow is only used to inseminate, to form the embryo. Embryos are taken, collected out non-surgically, without any surgery, by hands. Then these embryos are transferred, implanted in other cows. These cows will be acting as surrogate mother, and this cow is ready again for the one more procedure. This technique is what is known as multiple ovulation. In here, till this stage, FSH is given, multiple ova is released here. Till this technique, multiple ovulation. After that, fertilization takes place. One fertilization, when insemination, artificial insemination is injected, semen is injected into this cow. So fertilization goes on, formation of embryos takes place. Then that embryos are removed and implanted. That's why this technique is also known as embryo transfer, because you are transferring the embryos from the genetical mother to surrogate mother. You are collecting and transferring into different cows. Hence this technique is what is known as multiple ovulation and embryo transfer. By this technique, what is the advantage of this technique? Means to say that this is the significance of this technique. They may be asked also, what is the procedure of MOAT, and mention its significance. The significance is, in a short period of time, in a period of 9 to 10 months, here 8 animals or new animals are born, which are desired ones, which we needed from the selected breed of cow, selected breed of the bull. So in one gestation period, where normally naturally this cow can produce only one calf, whereas it has produced 8 embryos, and in these different cows, 8 embryos have been implanted. So 8 calf we are getting in a short period of time. This is a technique used to increase the number of the herd, size of the herd, to increase the number of the animals in a herd, in a group, in population. It can be increased in a very short period of time. This is a significance. And also, one more significance is the desired breed you can get here. Desired breed, because the semen is from the selected bull and the cow is a selected breed. So you get that. So this technique is a very important technique. Fine. Same technique is done in human beings also, because questions may arise now, because we have not done in our college 4th chapter, we have not done online. So most probably we will do that shortly. Right? So same technique can be done in human beings, where we call it as test tube baby. What we call it as test tube baby. Right? I hope it's clear. So let's moving on to the next very small topic. Right? After that, thereby we will complete this animal breeding. Then we have to start few small topics. Right? The next technique, this is the second technique what we have discussed in the breeding experiments. One is known as artificial insemination, the other is known as MOAT or SOAT. Second technique we have discussed today, the significance, the procedure, the technique, fine. And then the third technique, which is very much essential here for to assist these both techniques, is, fine, one is known as sperm sexing. What is known as sperm sexing. The third technique is, in my previous class I had done three techniques. Now this is the third technique. What does it mean to say that? It is known as sperm sexing. Sperm sexing. Don't get confused. You know our sperm is a gamete, male gamete, which has Y chromosome in it, whereas the ova is a female gamete, which has the X chromosome in it. Fine. Clear here? What do you mean? That's what this question I kept in your mind is in the previous class. You always see, most of the times you see only cows daily. Like for example, Holstein Friesian HF cows, Jersey cows. Only cows you see, but rarely, very rarely you come across these bulls. How is it possible to produce only cows? Because you get milk as a very good source of food for human beings from these cows. So only cows are being produced artificially. How is it possible? By this technique. What is it known as? Sperm sexing. What do you mean by this? Is to say that the sperms which are there in the semen, this semen contains both X carrying sperms, Y carrying sperms. Clear? When the ovum is fertilized by X carrying sperm, cow is born. When the ova is fertilized by Y carrying sperm, bull is born. XY male, XX female. In humans also same. For example here. So the X carrying sperm, semen consists of both X carrying and as well as Y carrying sperms. Now your experiment, you wanted to produce only females. In this technique we can talk as only cows, dairy animals you want to produce. So what has to be done? Only the semen which has only X chromosome has to be artificially inseminated. It has to be artificially inseminated into cows so that the resultant baby, fine, the resultant young one should be a female, should be a cow. So what has to be done here? Before artificial insemination, this X carrying sperms and Y carrying sperms should be, what has to be done? Should be separated. What has to be done? Should be separated. Now, the question comes, how it has to be separated? Because they are microscopic, in structure. You can't see them in the semen. So I have put one question here, microscopic. So what we use here? Microscope. What we use here? Microscope we use. Semen is taken, fine, based on their motility, based on their structure, size, fine, by using cell separating cytometer cell. This is also one cell. Sperm is a cell. Male gamete, male cell. Y is also a cell. Cell separating cytometer is the name of the device which is used under the microscope. By using under the microscope, semen is taken, suspended in a cytometer. Now you are observing from the microscope which is X chromosome, which is Y chromosome. So slowly you separate, you divide this Y chromosome separately, then X chromosome separately. When you are separating this from the semen, X carrying sperms are separated, Y carrying sperms are separated. When you use this Y carrying sperm, when Y chromosome carrying sperm fertilizes, the resultant young one will be bull. You know this. When the X chromosome carrying sperm fertilizes, the resultant young one will be a female or a cow. So in order to produce more dairy cows, fine, clear, HF, Holstein Friesian for milk production, Jersey and etc. like that, what we have discussed in exotic breeds. So only the X chromosome carrying sperms are used in case of artificial insemination. When they are injected into cows, only this cow gives rise to only cows. Now this cows again gives rise to more milk. This is what is the milk production is increased. That is what the concept of this chapter. Strategies for enhancement in food production. Milk is a basic food for human beings and other mammals. So here by using sperm sexing. What is sexing here? You are separating the gender. You are separating the male and female. You are separating the female and male and female gender. Y and X. That's why it is known as sexing. Gender is separated. That's why it is known as sperm sexing. Very simple technique. I hope it's clear. So here we complete artificial insemination, MOAT, multiple ovulation and embryo transfer, and also sperm sexing. These are the three techniques to increase fine breeding techniques. After this, very small topics we have. One is the pisciculture, fine, then apiculture, and after that we'll see one is about the fishes, one is about the honeybees. After this we can start plant breeding. I hope it's clear. Do repeatedly watch the class. Fine. Keep studying. God bless you. Good luck. Take care.